Hi, today I will be speaking with you about the influences of X and Y chromosomes on human behavior and looking at a comparison of psychopathology between XXY and XYY. So I'm a pediatric neuropsychologist at Children's National, and I collaborate with Dr. Armin Rasnahan's team at the NIH in the section on developmental neurogenomics. I imagine some of you listening may have participated and come through our study before the pandemic. So I have to start by crediting the contents of this presentation to Luke Schaefer, who was a research student in our lab and the lead author of the paper that this presentation is based on. He created many of the visualizations that I'm going to be using in the presentation, and he's the driving force and analytic mastermind behind this project. We're going to be submitting this paper to a journal in the coming days. Once it's published, we'll certainly alert folks and access to disseminate it to you all. I also want to point out a recently published paper from our lab entitled Deep Phenotypic Analysis of Psychiatric Features in Genetically Defined Cohorts, an application to XYY syndrome. This was led by our lab's principal investigator, Armin Rasnahan. And in this paper, Dr. Rasnahan does a deep dive into the psychopathology in XYY, and he lays out the analytic approach that Luke then applied to XXY for his paper. So in terms of the agenda, I'm going to be discussing why this research is important, as well as describing the approach that we used. I'll describe patterns of psychopathology in XXY, which I'm going to try really hard to refer to as Klinefelters for the remainder of the presentation, because I know it can get kind of confusing to hear XXY and XYY. Also, for the purposes of the presentation and to make it a little easier on everyone's eyes, when I'm referring to Klinefelters, it'll appear as XXY/KS. I'm then going to compare the patterns between Klinefelters and XYY, the patterns of psychopathology. And finally, I'm going to discuss how psychopathology in both SCAs is associated with aspects of everyday functioning, such as IQ, adaptive skills, and caregiver strength. So I want to review some terms and also go over why this research is important. So the term aneuploidy or sex chromosome aneuploidy, that can be abbreviated to SCAs, aneuploidies can serve as natural occurring models to study the effect of X and Y chromosomes on brain development and behavior. We look at the effects of the addition of sex chromosomes by comparing those with extra chromosomes to those with the typical XY karyotype. So on the left here, you see the typical XY male karyotype. And for this presentation, we're going to be specifically looking at Klinefelters and XYY. So this research used a genetics first or bottom-up approach. And what that means is that we specifically recruited individuals with particular genotypes. And for this study, it was those with Klinefelters and XYY. And we carefully looked at their brain anatomy and behavioral presentation by looking at neuroimaging data and having parents complete a comprehensive and thorough set of questionnaires to report on psychiatric symptoms. And then we also looked at what the clinical manifestation of the SCAs, or in other words, diagnoses, using gold standard diagnostic tools, and then looking at how these symptoms affect functioning for the individual themselves, as well as those around them. And we assessed that by measuring cognitive ability, adaptive functioning, and caregiver strain. Prior work and findings from our study are done by looking at groups of individuals with the same condition. But it's important to point out that these groups are comprised of individuals, and each person has their own pattern of strengths and weaknesses. There's a high amount of variability in psychopathology and cognitive ability between individuals with the same condition. And so a single individual's experience can get overshadowed by the overall trend of the group. So before I delve into the research that's been done and this the research that our group did that's in this presentation, it's always important to remember that just because we're describing group-based findings, it may or may not resonate or apply to you as an individual, or if you are a family member of somebody with Klinefelter's or XYY. So using the genetics first approach, it's particularly helpful to psychiatry because prior research that has looked at groups of individuals with Klinefelter's and XYY generally demonstrates that the addition of sex chromosomes increases psychiatric risk, creates vulnerabilities and aspects of cognitive skills, particularly language-based skills, and results in changes in brain anatomy. And the benefits of this is that it allows us to determine whether different SCAs can result in risk for different aspects of psychopathology, and whether the impact of increased psychiatric risk affects other aspects of functioning in the same or different ways across the SCAs. And a strength of our study in particular was that we looked at psychopathology not just through the lens of diagnoses, meaning is there a diagnosis or not? It's a categorical framework, whether a diagnosis is present or not. In addition to looking at diagnoses, we also looked at psychopathology using dimensional measures, meaning where does someone's score fall for a particular psychiatric domain along a spectrum. In terms of these study aims, what we were looking to do, we wanted to compare diagnostic outcomes and comorbidity within each SCA and across SCAs. And when I say comorbidity, what I mean is when there are two or more diagnoses present. But we also wanted to look at beyond just those diagnostic categories and look at patterns of psychiatric symptoms within each SCA and then compare these patterns across the two SCAs. And finally, we wanted to look at the relationship between psychiatric symptoms on daily functioning in the SCAs. So our current study used data from 102 individuals with Klinefelter's, and we compared those individuals to their own XY male control group. We also looked at Dr. Rasnahan's data from 64 XYY individuals compared to their own control group of 60 XY males. And that was for the comparison portion of the study, where we were looking at Klinefelter's compared to XYY. And parents in the SCA and the control groups completed 12 questionnaires, which yielded 66 different scales of psychopathology. And these were our dimensional measures. And within the SCA groups only, we also acquired additional data about psychiatric diagnoses using gold standard diagnostic tools. We also assessed for aspects affecting daily functioning in the SCA groups, including an IQ assessment, adaptive functioning assessment, and then a questionnaire assessing for caregiver strain. So initially, we compared each SCA group to its own control group on the 66 dimensional scales to determine whether the addition of extra sex chromosomes increased risk for psychiatric symptoms, and if so, which domains were particularly vulnerable. And then we compared how the differences in psychopathology between each SCA and its own control group compared across the SCA groups. In other words, were there certain psychiatric domains that were more or less vulnerable when an extra X chromosome was present versus an extra Y chromosome? So in terms of the diagnostic outcomes data, what we see is that there were high rates of comorbidity in both SCAs, and the most common number of diagnoses that was given was 2, meaning the most number of individuals had 2 diagnoses within both groups. And in fact, over half of the individuals in each SCA had 2 or more diagnoses. So these bars on the right represent all the individuals with Klinefelter's and XYY that had 2 or more diagnoses. And in terms of which psychiatric diagnoses were associated with each SCA, what we see is that almost 90% of individuals within each group were given at least one diagnosis at some point in their lives. and that neurodevelopmental diagnoses were the most common group of diagnoses in both groups. And this was largely driven by high rates of ADHD and autism spectrum disorder in both groups. When we look at which were the most common diagnoses within each group, on the left, we see in Klinefelter's, about 2/3 of the individuals were diagnosed with ADHD, about 1/3 also had diagnoses of autism, and about 1/4 of the 102 people with Klinefelter's were diagnosed with enuresis, which is essentially having difficulty with urinating not into the toilet. And then on the right, we see that the most predominant diagnosis in XYY was also ADHD and represented about 2/3 of the individuals, followed by enuresis in about 1/3, and then ASD was the third most common diagnosis in about 14% of the individuals. When we compare the diagnostic rates across Klinefelter's and XYY, we found that the group with the Klinefelter's group had statistically significantly higher rates of anxiety diagnoses relative to the XYY group. So this illustration here is demonstrating on the left side, these are the 12 different scales that parents completed about their children or young adults. And on this side, on the right side, these are the subscales that make up those scales. And so on the right side, these are the 66 total scales that we use to examine psychiatric symptoms. All right, so initially we're going to look, these are the dimensional measures, so we're going to look at psychiatric symptoms in Klinefelter's first. So I'm going to take a moment to explain this graph that looks really complicated, I'm sure, but I promise you it's not as complicated as it looks. So what this graph represents is, try to get a laser pointer here, along this left column here, these are those 66 dimensional scales, and they've been ranked by least different from the XY male control group to the most different from the XY male control group, meaning the scales that are least impacted in Klinefelter's to most impacted in Klinefelter's. And then on this bar over here, this is representing effect sizes. Effect sizes is basically how clinically meaningful is the discrepancy that we're seeing. And the more saturated the color we see, the higher the effect size, or the more clinically meaningful that difference is. And if you're seeing 2 asterisks, that just means that discrepancy between the Klinefelter's group and the control group is statistically significant. So when we look at which were the scales that were most impacted in Klinefelter's, we see that the greatest impact compared to the XY control group was observed on scales measuring autism-related traits, social difficulties, and mood and anxiety symptoms. We see intermediate levels of discrepancies for attention problems, motor coordination deficits, and disruptive behaviors. And finally, there are minor, often statistically insignificant elevations seen in scales assessing obsessive compulsive features, aggressive behaviors, and antisocial tendencies. So now we're going to do the same analysis or the same discussion, but for XYY. Again, this is the same type of graph I showed you on the previous slide, where the left side here represents the 66 scales, and they're ordered by those that were least impacted in XYY to those that are most impacted in XYY. What I would like you to note is that all of these bars here are shifted further to the right than they were on the previous slide showing XYY data. And similarly, these bars up here are showing deeper saturation. So essentially what we're seeing is that the scores in XYY are much higher compared to their own control group, and that those differences are much more clinically meaningful because the effect sizes are larger. And so that's something to keep in mind when we're thinking about a comparison between XYY and XXY. So now when we look at which specific domains of psychopathology are more elevated in XYY compared to their control group, we see, as in Klinefelter's, the highest elevation is seen in social difficulties and autism traits. We see intermediate levels of elevations on hyperactivity, inattention, externalizing behaviors, mood and anxiety symptoms, and repetitive behaviors. And the smallest levels of elevations are observed in obsessive compulsive symptoms, aggression, and antisocial tendencies. So now we're going to talk about the relationship between those psychiatric symptoms and aspects of daily functioning. So really, what is the relationship between psychiatric symptoms to IQ, adaptive functioning, and that really just refers to completing chores, self-care, personal hygiene, getting around in the community, as well as caregiver strain. And the latter, the caregiver strain, was assessed through a questionnaire. All right, so this graph here is similar to the one I showed you before, but in terms of looking at the relationship of psychiatric symptoms to aspects of daily functioning, this bar on the left represents those effect sizes, looking at the impact of those scales and how different they are compared to XY controls. This first column here is showing the relationship of each psychiatric scale to the relationship to IQ. The second bar here is showing the relationship of each scale to adaptive functioning. And the third column here is showing the relationship of each scale to caregiver strain. So when we look at IQ and its relationship to all the scales, there's really only one scale that comes up as being significantly associated with IQ, and that is a learning problem scale. With respect to adaptive functioning, we see scales assessing social, attention, and autism-related differences were most strongly associated with adaptive functioning. And scales assessing autism traits, social difficulties, externalizing, and aggressive behaviors were most strongly correlated or associated with caregiver strain. I think what I want to be sure to highlight here is that the scales that are most impacted, meaning those that are at the top of this scale here, aren't necessarily those that are most linked to some of these daily functioning outcomes. So, for example, these scales that are down here that aren't necessarily as impacted in Klinefelter's compared to their control group, they're still having a significant impact on daily functioning with respect to adaptive functioning and caregiver strain. All right, so now we're going to walk through that same type of analysis for XYY. As with the Klinefelter's group, learning problems was also associated, or the most associated with IQ in XYY. And almost exclusively social and autism-related scales were associated with adaptive functioning. And autism, social, externalizing, irritability, and hyperactivity scales were associated with caregiver strain in XYY. And again, as with Klinefelter's, we see that the most impacted scales are not necessarily the ones that are most linked to some of these outcomes, such as adaptive functioning and caregiver strain in XYY. So now in terms of how to bring this all together and conclusions to take away from this, I think it's important to remember that having an extra X or Y chromosome in males does increase risk for most psychiatric diagnoses. And in particular, individuals with Klinefelter's are at a higher risk for anxiety disorders compared to those with XYY. Comorbidity or having more than one diagnosis is extremely common in SCAs, particularly Klinefelter's and XYY, which is what we studied in our project. And in fact, more than half of each of our SCA samples had 2 or more diagnoses. And it's important to not just look at psychiatric health through the lens of diagnostic criteria and whether or not someone meets criteria for a diagnosis, because a lot of times they could be subclinical symptoms that are present that are nonetheless contributing to impairment or affecting daily functioning. And in fact, we observed that some of those scales that weren't that discrepant in XYY and Klinefelters relative to their XY control groups, some of those less impacted scales were nonetheless very much associated and linked to things like adaptive functioning and caregiver strain that affect daily functioning. So I want to take a moment to thank all of the participating families that came through our study, as well as AXYS for their help in recruitment and allowing us to disseminate the findings at the conference. This is a recent picture taken of our lab. Not all the folks that are named over here are in this picture because many of the people that contributed to this project are no longer in the lab. They've moved on to bigger things and moved on in their careers. But nonetheless, we wanted to thank them given their contributions to the project. And if anyone has any questions, I am available to take them now.